All right, we're right around 10 o'clock, and so I'm going to go ahead and get started. Thank you to everybody for joining us today. This is our Cape Perpetua speaker series, and we host this every Saturday at 10 a.m. this year, November through March. Today, we are going to hear from Doug Glavich on lichens and lichen bioindication science. And next week on December 12th, our presentation is going to be on the 5 Mile Bell Landscape Restoration Project. Real quick before we get started, my name is Tara Du Bois. I am the communications coordinator for the Cape Perpetua Collaborative. And our goal is to foster conservation and collaboration within local communities. And our 3 guiding principles are community engagement, leveraging resources, and engaging in partnerships. And as you can see at the bottom here, these are our founding partners, and we have a variety of partners, including federal agencies, state agencies, some local nonprofits, and the Confederated Tribes of Coos, Lower Umpqua, and Siuslaw Indians. Our focus is around the Cape Perpetua Marine Reserve and that land-sea connection. And I always like to share a fun fact, and today's fun fact is the Cape Perpetua Marine Reserve research began in this marine reserve in 2012. Harvest restrictions began in January of 2014. And at the time, there was moderate fishing pressure on ground fish in the rocky reef habitat. And high fishing pressure on crab and soft bottom habitat. And you can find out more information about the Marine Reserve, about the collaborative. And as you can see here, we host a variety of different events, community science events. Some are seasonal, some are monthly. So you can check that out at our website, capeperpetuacollaborative.org. We've got an events tab where if you're looking for events, we've gotten a calendar up with everything that we've got posted. I will be in about 2 weeks listing out our presentations for the beginning of the year, January through March. We are also hosting a Young Scientist webinar series on the second Tuesday of every month at 5:30 p.m. And our next one is coming up on Tuesday. December 8th in the evening, and that will be around the intertidal and sea star wasting disease. So again, check out our website for more information on those things and to get registered. And please feel free to connect with us on our Cape Perpetua Marine Reserve Facebook page and our YouTube channel. We will be recording this presentation as well as all of our other presentations, and that's where we'll be hosting them, is on our YouTube channel and on our website. So if you can't make it, there's usually a replay available. And with that, I'm going to introduce our speaker, Doug Glavich. He's an ecologist for the Forest Service and Northwest Oregon Ecology Group, and he has worked in lichen ecology and lichen bioindication in the Northwest for nearly 20 years. Doug, it's a pleasure to have you back. You've presented for us before in this speaker series. And it's very sought after topics, and I really appreciate you coming back. And thank you to all of those who have registered for previous presentations with Doug, particularly last March when COVID started. We had to cancel, regroup, and reschedule, and so I really appreciate all of the adaptability. So with that, Doug, I am going to stop sharing and you can take it away.
Thank you, Tara. All right, let's see. Is that coming through okay?
Yes, it is.
Okay, excellent. Yeah, like Tara said, yeah, I've spent, nearly 20 years in lichen ecology and in some bioindication projects, even though these days I kind of focus on meadow systems, but yeah, so I'm just going to kind of share, yeah, sort of some lichenology with you and some of these projects I've worked in. Yeah, if I can, I just got to get this thing to advance. There we go. All right. So the way I kind of set this up is sort of like a lichenology 101. And it's just to kind of to provide some brief lichen basics, kind of what these things are, how they function, kind of get everybody on the same page, because I think there's a variety of experiences out there with lichens. And we'll take a look at some selected lichens of the Coast Range, some that are common, that if you're just hiking around out there in the forest, you'll see them, and maybe some less common or rare that you might go looking for. And we'll take a look at some of these, a brief look at some of these lichen bioindication projects I've been involved with. So first, what is a lichen? It's really a composite organism. It's kind of a symbiosis of a fungus and a photobiont, which is the photosynthetic partner, which is either an alga or a cyanobacterium, and sometimes both. And basically, the photosynthetic partner makes sugar, feeds the fungus, and then the fungus in turn provides a protective housing. And then there's new work that suggests there's a third partner, yeast, and we're not totally sure what its role is in that partnership. And of course, you often hear cyanolichens. Well, lichens that have a cyanobacterium as the photobiont are often referred to as cyanolichens. And lichens come in different forms. So there's two broad forms. There's the microlichen, which is your crusty types. Those things are, that you see plastered all over tree bark and rocks. And then there's macrolichens, which are your more sort of three-dimensional forms. And those are broken down into fruticose, which are shrubby or hair-like lichens. And they tend to have that algal layer wrapping around the branches. And then there's the foliose forms, which are your leaf-like lichens, and they have an algal layer that's only on the top surface. And that becomes important because sometimes you see lichens that look shrubby, but they actually have kind of like skinny, flattened branches, and they have and that becomes important if you're looking into getting into identification. So those are your basic forms. So how these things reproduce? They do a lot of cloning. The hair-like lichens, they produce often through fragmentation, where just little pieces of the branches break off and then form a new lichen somewhere. And then many lichens have actual reproductive structures, or like propagules. And here's couple of examples: Ceridia, which are sort of these bundles of fungal hyphae and algal cells, and they kind of look like granules or powder on the surface of a lichen. And then there's Acidia, which is kind of a projection that has the cortex or the skin kind of covering these projections, and they just sort of break off and form a new lichen. And then they also produce by fungal spores. And of course, most of the fungi that are associated in the lichen symbiotic relationship are ascomycete lichens. And so you'll see mostly these ascomycete fruiting bodies. And so these fungal spores, so that's just the fungus. And so it must find free-living algal species on the landscape in order to sort of germinate and develop into a new lichen. And here's sort of a few notes on just some real basic physiology or adaptations. Lichens, they basically absorb nutrients and moisture from the atmosphere. They don't really have any, they don't have any cuticles or roots to regulate nutrients or water, and so they just absorb everything. They're dormant when they're dry, so their metabolism kind of cranks up when they get wet. And species are differentially adapted to climate and moisture regimes, or climate zones, for hot and dry, cool, wet, for example, and they're differentially adapted to atmospheric nutrients like nitrogen. So a bit on cyanolichen physiology. It does differ a little bit from the green algal lichens. So the cyanobacterium partner requires liquid water to activate the lichen metabolism. So, and green algal lichens can they can activate at much lower water content. So it just takes a little bit of humidity in the air to often just get a green algal lichen cranking, but the cyanobacterial lichens or the cyanolichens have to get pretty wet. So they're typically found in more higher humidity environments like riparian and wet microsites like embedded in mosses. And the cyanobacterium partner also fixes nitrogen. So cyanolichens can also be an important forest nutrient input. And for the identification of these things, if you want to get into ID, you want to pay attention and learn these reproductive structures because they become important. Besides things like lobe size and shape, it might be the present shape, color, and position of Soralia, those granules, those propagules. They'll either be, like laminal, like across the surface, or sometimes marginal. And those ascidia or projection structures, they can be globular or branched or cilia tipped and many other shapes, and those are important. And then in some cases where there are lichens out there that are basically identical to the eye. And so those you can really only separate by their secondary chemistry. And there are quick and dirty spot tests that you can identify that chemistry and separate the species. And they're just sort of color reactions where you dab a little bit of bleach or potassium hydroxide and some other agents to the interior or the exterior of the lichen, and the color reaction tells you whether it's one species or the other. And then of course, then there's thin layer chromatography, and that's in more rare cases, unless you're a taxonomist, where you've got one of my roles in one of the bioindication projects for many years, I identified about 3,000 lichen samples per year, and about 5% of the hardest genera required thin layer chromatography to separate them out. So it's a pretty small amount of cases. And then there's spores. The macrolichens require less spore work. It's pretty routine in the crusty types. So if you get into crust lichens, it's a lot of microscope work and takes a little bit more time. So looking at some of the environmental adaptations and part of the lichen ecology is that they are, as we mentioned before, that they are sort of differentially adapted to sort of these different moisture regimes, and even at the fine scale or the microclimate, and this is even within a single tree or the canopy of a forest stand, where you've got lichens that are adapted to, say, living on the outer crown, where they get they experience more drying, and so they're wetting and drying a lot. You have species that are kind of out in the middle of the crown on branches where they're having kind of maybe a middle moisture envelope, and you have some that might be in a wetter site on the tree where you've got a lot of water draining on the trunk. And so you have these lichen species that are adapted to all these different positions in the tree, in the crown or canopy. And of course, then that might get you thinking that, well, if you have a tree with a much more diverse structure, like an old growth tree, you might have a diversity of microsites and thus a diversity of lichen species. And yes, that's the case. Older trees and older forest stands and more complex canopies have a much higher species diversity for lichens. And then at the broad scale, environmental adaptation, lichens are differentially adapted to particular climate zones at the landscape level. And that might be a desert or a subalpine or maritime, for example. And we'll revisit this a little bit in the bioindication part of this talk. And so now let's kind of dive into lichens of the Coast Range. And I'll sort of apologize right now for my use of scientific names. Common names have always been sort of challenging in the lichen world, but where I could find them, common names, I included them on the slide. So Loberia oregana, it's really common in the Coast Range and throughout the Western Pacific Northwest. This lichen actually has both a green alga and cyanobacterium photobiont. So kind of the best of both worlds. If you cut open a lobe and cross section, you'd see that green algal layer and these pockets of cyanobacteria. And this species has been found to be an important nitrogen input in some forests. And these species range from like the size of your hand, and I've seen them the size in old forest as like almost trash can legs. So they get quite big. And Sphaerophorus, there are a couple of species. This is probably one of the most common lichens in the Coast Range. And sometimes you might walk into a forest stand and that's one of the only macrolichens you see. And so you'll just see a lot of Sphaerophorus kind of running up large conifer trunks. And the genus Usnea, this is an interesting group. Yeah, they're all or mostly epiphytic, and probably all epiphytic in the Coast Range. The distinctive character of the genus Usnea is that central cord. There are other kind of yellowish sort of beard-like lichens out there, different genera, but this is the only one with a little kind of a mini bungee cord on the inside. So if you kind of crack the lichen open and kind of stretch it out, you'll see a little cord in there, and then you know you've got the genus Usnea. And it has both shrubby and pendulous forms. Usnea cornuta is a very common Coast Range species. There are lots of other shrubby species out there, and they are a little bit challenging to ID. The differentiating characters are subtle and often require chemistry. And Usnea longissima is one that you've probably seen. It gets really abundant in small Coast Range valleys. So if you're driving up, like out through the Alsea River or out Highway 20 and you get down along floodplains where there's like large big leaf maples, and you see these big garland-like lichens draping on the tree, well, that's always going to be Usnea longissima that forms those large garland-shaped lichen forms. And it is scattered across the conifer forest and in the Coast Range, and it does gain some abundance as well in older stands. The tube lichens or Hypogymnia, I think this is a really cool genus. These they've all formed these hollow tubes. There are quite a few species out there, and these are some examples of common ones. So the inactiva, if you're out there and you see this almost like perfectly dichotomous or forked branching, in the Coast Range, it's most likely inactiva. Enteromorpha kind of has this sort of melty look that's sort of oppressed and kind of a puffy, melty look. It's sort of oppressed to the branches. It's most likely Enteromorpha, although there is a nearly identical one that you can separate by chemistry, but I think this is one that's more common. And Physotes, I think, is really cool because it has those exploded lobe tips. And you can see that the lobe tips are lined with the ceridia. And as you recall, the ceridia are those bundles of algae and fungal partners that are the propagules for reproduction. And as I mentioned, they are also, as you see, important in identification. So that's a character of that particular Hypogymnia, the exploded lobe tips with ceridia around the edges. So you'll most likely see these when you're out hiking around. Of the specklebelly lichens, Pseudocyphalaria crocata, I think, is kind of one of my favorites because it has yellow ceridia. And there's quite a lot of it in those round patches across the thallus. And it's a cyanolichen, so it's found, in kind of damp microsites and riparian areas. And one similar one that I needed to showcase is Pseudocyphalaria perpetua, because it was first discovered in the Cape Perpetua area. It's much less common than crocata, but it is out there, and it looks a little different or slightly different in that the yellow ceridia are mostly on the margins instead of in those round patches across the surface of the lichen. So if you're out hiking around the Cape Perpetua area and beyond, keep an eye out for that one. And Peltigera. You see these out on the forest floor, and you've probably seen these if you're out hiking out in the Coast Range. And these are a couple of common ones out there. And the Peltigera genus, what distinguishes them is they have venation on their lower surface. Peltigera britannica is one of the common sort Peltigera that's got both green algae and cyanobacteria, and those black dots on the surface of the lichen are actually the cyanobacteria pockets. And then Peltigera membranaceum is one of the common kind of brownish gray ones that you see on the forest floor. And those kind of orange tips that you see are the actual apothecium, or the fungal fruiting body. Plasmatia is a very common genus. And Plasmatia glauca is probably one of the most common lichens in the Pacific Northwest. You can even see tiny amounts of it on the east side of the Cascades out in relatively wet sort of microsites, but gains abundance on the west side. But the one I wanted to showcase here is really Plasmatia lacanosa, as it's a coast range Plasmatia. It's fairly uncommon and found in riparian areas, even though it is a green algal lichen. And it has that very strong ridging. So you see pale greenish, kind of greenish bluish gray lichen with that strong ridging. You've probably got lacanosa. So we just kind of visited some lichens that are kind of broadly across the coast range, but we'll just take a little bit of a dive into the hypermaritime species. And these are those that have a strong, that require a strong ocean influence. And these are, on forest edges inside of the ocean and in the dune forest. So Ramalina menziesii, this lace lichen. So when you're out, especially in the dunes, right on those forest edges, and you just see this, it's almost kind of this almost beard-looking lichen that's just really abundant and dripping off the trees. It's probably Ramalina menziesii. And you can tell if you kind of separate open the branches and look for the reticulate netting. And this also has a bimodal distribution because it also occurs in large inland valleys, like the Willamette Valley. Here's another tube lichen, but this is restricted to the coastline. It looks similar to one in a previous slide for the interior of the Coast Range, but if you're out on the coastline and you see one looks like it's heterophylla, noted by its irregular side branches, but if you're out on the dunes, especially, you'll see this in open pine forest. There's a lot of it on the beach pines. And again, out on the dunes, this maritime reindeer lichen, or Cladonian portentosa, you find it on the ground, and it's pretty showy on kind of moist or wet days, where it kind of almost looks like snow patches. And you often find it kind of embedded in with that sort of matte manzanita on the dune forest floor. The horsehair lichens, or that Bryoria group, there's quite a few Bryoria out there. I needed to showcase this one because I've done a lot of work on its ecology and taxonomy. And this particular lichen is, even though it gains some really high abundance in some areas, and especially dune forest, it's actually quite rare. It really only occurs from about Florence, Oregon, south into Northern California. And what's distinctive about this particular horsehair lichen, or this, it's now Sulcaria, is that if you look at it with a hand lens, the branches have kind of distinctive spiraling stripes around them. And the last lichen we'll look at in this series of checking out Coast Range lichens is this Tilischistes flavicans. This one is really rare in the Northwest. It's actually quite common along the coast from Central California south, and I think as far as into Mexico. But in Oregon, there's only known from a few locations, and only on headlands in very exposed areas. So if you find this, I recommend not collecting it, but maybe reporting its location. So now, let's take a dive into bioindication work. So, how do we get from lichens into some output like this? Well, we're going to take a a look at Forest Service Lichen and Air Quality Monitoring Project for Oregon and Washington. Spent a lot of time on that. And climate indication potential, and which will be, a component of those broad scale climate environmental adaptations. And we'll take a quick look at a project that assessed semi-volatile compounds in the western US, and one looking at heavy metals in northeastern Washington. So the layout for this big sort of Forest Service Lichens and Air Quality Monitoring Project. So there's a large plot network out there for the Forest Inventory and Analysis Program that measures forest health. We use that network that's got over 3,000 plots across Oregon and Washington, and also added supplemental plots in urban areas and coastline and valleys and places where we need to complete environmental information. And on these plots, we measured or surveyed the lichen species community of epiphytic macrolichens and their abundance, and also collected lichen tissue of ubiquitous species, usually 1 or 2 species per plot of something very common across the Northwest. And those were sent to a lab to analyze for nitrogen, sulfur, lead, other metals, and other elements. And there's a plot diagram that kind of illustrates the survey area, which is roughly about an acre survey area in a plot where all that information was recorded or collected. And the monitoring, this was initiated in the mid-90s, resampled every 10 years. And I think the 20-year sample might be complete. If it's not, it's nearly complete. And just a bit on the lichen community, just so we all know what I'm talking about. And it's a group of species living together in a co-adapted environmental neighborhood. And as we'll see, that tends to be precipitation, air temperature, and atmospheric nutrient content. And also recall that lichens are more a reflection of climate, air, nutrients, because since they can't regulate their metabolism or water with other structures, whatever's in the air is in them. And why epiphytic macrolichens? So why don't we bring in the crust or the terrestrial or species on the ground? And that tends to be just because the epiphytic macrolichens, it's a distinct group that's quickly surveyed, relatively easy to ID. And if you add crust or everything on the ground, then that becomes sort of a daunting, and the amount of work would be, it would just be too hard to do. And epiphytic macrolichens have demonstrated repeatability and also illustrates environmental gradients. So we won't dive too much into this, but just kind of wanted to showcase how some of the other output is generated. This is an ordination, and it's a very common ecology or ecologist analytical method, because what it does is it analyzes or correlates groups of things, particularly species composition, with environmental gradients. And basically the only thing you really need to know on this is that we've basically defined the axis, like axis 1 as high air quality to low air quality, and axis 2 is a climate, which is warm winter to cold winter temperatures. And in each one of those dots is a plot with a species composition list. And so where those plots line out against those gradients, they get scored based on the axis value. And so, yeah, there's a lot to go over on this kind of stuff. You can spend a full hour just on this. But if there's questions on this, we can maybe get to those later. But that is how these plots are scored. So you get these scores from that ordination method, and these scores can be grouped into these different climate or air score groups. And then you can actually you can interpolate them into maps. And so you can get from best to worst, air quality if you're grouping these axes values. And just, there's always questions about the sources. And so sulfur source, and this, of course, this air quality map is mostly sulfur and nitrogen influenced. And so sulfur sources are usually coal-fired power plants. Nitrogen sources are often agriculture and vehicle emissions. And also note that while you have these really degraded and polluted areas, it's not necessarily the health or the air quality index is not necessarily polluted to sort of maybe human health standards. So the emission sources are usually in compliance, but these are really poor air quality for lichens. And so they do tend to be hazardous to lichens, but do meet human health standards. And of course, if you are interrupting the lichen species composition, then that is affecting ecosystem processes. And for monitoring, when these plots are resampled in these decadal survey timeframes, the data can then be re-entered, and these plots can then be rescored through that ordination process. And if there's any environment influence that shifts the species composition, then those plots will get different scores. And what it might look like for species composition in sort of the worst air quality sites, you're just sort of left with these very tolerant lichens. So remember that lichen species are differentially adapted to different nutrient loads, and so there are some lichens that tolerate it. So in those areas, you get lichens that are, you only get the tolerant species, and then in the best air quality, you get all the sensitive species. So that is in part what drives the model. And also sometimes we want to link what we're seeing in the lichens to what's actually happening on the ground. So we've done some work like have these field sites where we collect lichen tissue that then gets sent out to the lab for analysis of those elements, including nitrogen, and also set up precipitation collectors that have ion exchange resin tubes that extract elements from precipitation, both from canopy throughfall and wet deposition. And canopy throughfall there, it says wet and dry deposition. That's because any particles that are depositing out in dry times, they're just collecting on the surface of the canopy. Then it gets washed off with precipitation. So then you get a mix of wet and dry deposition. And then we can take that data and we can correlate. The percent content of nitrogen that's in the lichen tissue with, say, nitrate in the canopy throughfall. And nitrogen concentration of lichen with total nitrogen. And here's an example of taking that information and relating. Well, we know now when we have 1.5% nitrogen in the lichen, we've got 7 to 11 kilograms per hectare per year depositing out in the landscape. And this climate is part of that same model output. And so climate indication kind of gets again at that broad scale environmental adaptation, the kind of a landscape level macroclimate. And so you've got species composition or lichen communities that are adapted to these particular climate zones. So you've got these species that are adapted to high elevation moist. So these are species composition that you'll see up like right along Cascade Crest or up probably above 4,000 feet or so, 4,000 to 5,000 feet. You have mid elevation cool moist, kind of your mid elevation mountain slopes. You have your low elevation warm moist, and then you've got these dry types. You see that on the east side throughout Sisters and Bend and the Ponderosa stands. And then of course, lichen species composition you'll see on the immediate coastline. So you see there's potential there for some lichen indication work for climate. I've done a little bit of work on climate assessments in the Cascades, doing some modeling at different climate parameter optima. And here's an example lichens of cold winter environments like this Carnophylltia, you know, their sort of optimal abundance occurs at like 14 to 21 degrees in the winter. And you've got warmer species where they occur at 28 to 36 degrees in the winter. So you can see that lichens have these particular climate zones that they live in. And so that's kind of setting the stage for potential for climate indication and change through monitoring. If you've got species from, you know, different climate zones moving into different locations. And kind of what we know overall, this is just a quick example of a couple of comparative methods where European scientists came over and compared methods here in Oregon across the entire state with lichen data using the European method in the United States, and they basically came up with roughly the same answer. So we know that kind of almost no matter what method you use, you basically get precipitation, relative humidity, and temperature, and of course, and nitrogen are the biggest drivers across large landscapes. And knowing that those methods are comparable, then we can probably get at maybe some global scale climate or even other indication out there with combined methods in different locations around the globe. So that was that Forest Service and air quality project. Here's another one I just kind of wanted to do a brief look at, and it's this is the Western Airborne Contaminant Program that looked at semi-volatile organic compounds in the Western US. And semi-volatile in that these are compounds that deposit on the landscape, temperature warms up, they volatilize, air temperature cools down, they deposit out again, warms up again, they volatilize again. They kind of bounce around the landscape. And so to get an understanding of this across the Western US, there's a multi-agency effort. And of course, the national parks were a convenient sampling location. And there were lots of fun helicopter rides around the Arctic. And there's a photo of me weighing bags of lichen in Sequoia Kings Canyon National Park. There's lots of backpacking trips through all these areas. But one of the things that we found, here's an example output from that work, sort of assessing pHs through with lichens, is the Glacier National Park was the winner for the highest concentration of pHs, and turns out that was correlated with an aluminum smelter in Montana. And so there's a very large report on this that has the results from all of the different resources analyzed, and so there'll be a link posted at the end. So anybody who wants to find that. And then there's this heavy metals project that we started. Some of our monitoring plots indicated there were some elevated metals up in the upper Columbia Basin in northeastern Washington. And so I worked with a graduate student to go up and do some more sampling to get an indication of the distribution of elevated metals and up in the upper Columbia and especially in the Colville National Forest. And yes, he did find through lichen tissue analysis that we had some pretty high numbers, 294, 268% above background lead, for example. And there's been a long history of mining in the area, and there was a smelter that across the border that was polluting the area from the early 1900s into the 90s, and then they did clean up. But we wondered if the source was still, if that was still the source. And so I partnered with the Scheel Lab at Oregon State University, and Dr. Scheel is an isotope chemist interested in environmental monitoring and with lichens. And so she took her students up to Canada and sampled around Trail BC near the smelter and vicinity, combined it with our sample and found a concentration gradient of metals. And here's an example of the lead. And not only that, but was able to identify the isotope signatures in the metals and the processing signatures of that particular smelter. So we were able to identify that, yes, that smelter was still depositing out metals on the landscape, though at thresholds, way beyond background for the lichen, but still probably within compliance with human health. So that there sort of concludes most of the bioindication work and the lichenology stuff, but I wanted to leave you folks with some resources if you get interested in the lichens. The Forest Service Lichen and Air Quality Database website has all kinds of lichen information, but I think the database is particularly useful. If you want to map lichen species on our plot network, then you can do that. And if you want to say, if you want to plot out pollutants or different elements and see what if they're above background or not, and you can see that glowing red up in northeastern Washington, the plots that were above background for lead. So yeah, you can perform these right online. You can download the data. You can map these things out, and I'll post a link to this as well. And of course, some identification resources. The second edition macro lichen guide is the guide to use. And then I recently found this online key. Seems promising, though I haven't used it yet. And of course, the coffee table book, lots of great photos and distribution maps. So anyway, I just want to thank everybody for showing up and want to hear something about lichens. There's some links there for some of those resources. And I guess if there's time for questions, I can take those.
We did get some questions rolling in.
Right.
I want to just mention real quick, one of them was about they were able to join late. And so I just wanted to let them know this is being recorded and it will be posted probably by Monday afternoon at the latest Tuesday on our YouTube channel and our website. You'll get an email with the links to those as well on Tuesday. So look for that. And then the first question for you, Doug. The horsehair lichen coloration seemed brown and another rare one was orangey, yet both descriptions include from green algae. Can you please explain the color differentials?
Yeah, the color differentials, you'll find through lichens in general that they'll have lots of different color, their exterior color will not oftentimes the algae will, or their photosynthetic partner won't necessarily play a role in their coloration. So even in that particular horsehair lichen, you might even see within that same species, you'll see a color variation from say like a palish, kind of a real pale grayish brown to kind of a dark reddish brown. And those are usually due to secondary compounds in the skin or the cortex of the lichens that provide the coloring. And oftentimes those come as, sometimes those act as sunscreens, for example, for blocking UV. Might be one example, but yeah, the coloration usually is secondary compounds in the cortex.
And then another one, those beard lichens with the bungee cord, they forgot the name, and I don't remember it either. Do they grow on dead tree branches or do they kill live branches?
They will grow on both live and dead branches. They don't kill trees. They don't have any, none of their sort of fungal or other of their tissues, they don't reach into the wood at all. They're attached. So yeah, but they will take advantage if there is like a dying branch of a tree, it will often sort of populate up with lichens. So lichens will take advantage of that surface, but yeah, they don't kill the tree.
I've always wondered that. A lot of people mention that, like when they're driving or they're coming in, they're like, as we were coming in, we saw all this stuff.
Yeah, it happens a lot. You'll see, like sometimes you'll see like a snag top or some branches die off, and the lichens will colonize it because they'll stick to
Wow.
the
Wow.
surface. Yeah.
Is it possible to determine how long the exposure to metals has been as opposed to the concentration time period?
Um, I would say we haven't quite gotten to time period exposure yet, although there needs to be a little bit more work on how long, because eventually um, compounds that lichens absorb, whether they're metals or other things, they do eventually leach out. They will bind and store for a while, but probably not long term. And then depending on how long those lichens have been around, because those, a lot of those lichens have probably not been there for, some of those lichens have probably not been there for, 50 or 60 years. They're probably younger than that. So we don't quite have a time period yet, but we do know that the lichen, like the metals that we're finding in the tissues of the lichens at the time they're analyzed, they're probably fairly recent. They probably were absorbed, somewhere in within a few years to, yeah. So we don't totally know, but not long-term storage.
Very cool. Are the winter temperatures versus summer temps the main limiting factor for lichen geographical locations?
In part, yes. Yeah, winter and summer temps both play a role. Usually when we run these models, maximum summer temperature comes out as well, but winter temperature comes out as one of the strongest, but they definitely are, at least in the west. Now, when you get onto the east side, the model that I showed was the west side model. There's actually an east side model as well. And precipitation and moisture availability comes out as a higher driver on the east side, whereas the west side temperature is a higher driver.
And then on the websites that you had mentioned, is there additional food chain information?
There is some. There is some information on there, and there's lots of interesting stuff on lichens in the food web and sort of wildlife use of them. For example, I think this is probably happens more on the Cascades, but flying squirrels, lichens are part of the winter forage diet for flying squirrels, which are in turn important for spotted owls, right? But the particular lichens that the flying squirrels eat are probably in the higher elevation parts of the Cascades, probably on the edge of the owl and squirrels territory. But I think on that website, if you look around, there's lots of information on there. So there could be some wildlife stuff. And if folks are interested in particular topic areas, they can always send me an email. And if I've got some papers on it, I'll send them their way.
Very cool. Do any lichens damage or kill trees?
Not that we know of.
And then this one, I know lichens are symbiotic and won't kill the tree, but if we see lichens on a stem of a tree, does that indicate it isn't as healthy, or do they grow on healthy branches and trunks too?
They grow on healthy, well, there's both. There are lichens that are adapted to both probably dead wood and bark. So I think there's actually some papers out there that separate out. Here's a group of lichens that just grow on dead wood, and here so I think there's some papers out. I don't remember which ones, but there are. Yeah, I think there are some species that, and I don't know if it necessarily would indicate that the tree is dead. There's probably some other indicators that are quicker to identify a dying tree, but
right.
Yeah.
Do live plants produce compounds that inhibit lichen growth?
Yeah, I would say yes, and it might be species. I mean, of course, there's some out there like real obvious ones that are not native to here, but like eucalyptus is one.
Oh.
And sometimes I've seen this on redwoods. where I've seen there are lichens, obviously lichens grow on redwoods, but I think on some places of parts of the bark, there's lots of species that I don't see there, and there could be some compounds in redwoods. I'd have to go back and look at the redwood and lichen. There's lichen communities associated with redwoods, and so there's probably some species that might be deterred from that particular chemistry and that are in redwoods. But as a rule, yeah, I mean there are some, I think, but I'd have to look into it.
And the questions are continuing to roll in, so I'm just going to keep going through them for you.
Yeah, keep going.
Okay, this is one I hope I can pronounce properly. Are the cyanobacteria that lichens have similar to the poisonous lichens that occur in water bodies?
Oh, okay. So, yeah, because I think what they're referring to is that you get those kind of cyanobacterial blooms in lakes that are hazardous.
Oh, right.
And so, and of course, those are with the cyanobacteria blooms that you see in lakes. I would have to say that I don't think it's the same. As far as I know, it's not the same species of cyanobacteria that form in that lichen symbiosis.
Okay.
Yeah, I don't think it's the same species.
And then based on what you know, can you forecast the effects of climate change on lichens?
Yeah, I think it can. And kind of why I say the climate monitor or the climate indication potentials, because the work is really starting up. The work has been really focused on air quality for a long time, but now as the climate issue has reared its head, and I was starting to analyze the data for climate, and from what I've seen, I wasn't able to spend the time validating yet, but I think I was seeing a few species shifts. So I think you will be able to see once this 20-year data set is analyzed fully. I think we'll be looking to see if there's been any species shifts in there. So I think we'll see it.
Very interesting. Is there a difference in appearance in dormant lichens versus active lichens?
Well, I would say yeah, a little bit, because their color will change. So, in the summertime when you're out there and it's dry, the lichens their coloration won't be as vivid. They'll kind of be more pale. And so I would say their overall color is improved when they're wet. So they will, when they're dry, they'll be a little more pale looking.
Do rock lichens have any adaptations to toxins emitted from rocks, such as selenium, peroxide, etc.?
That is, I haven't done a lot of work in the microlichen world and in the rock lichen world, but from my little bit of reading on it, I think it's the case, and I suspect that you'll probably find species that might be even associated with particular geologic types, like serpentine, for example. And I know that there are some lichens that are lichens that are, specific to like limestone, for example. So I think you might find those that there are lichens that are not deterred by particular chemicals, and some that are.
Okay, you mentioned that some lichen provides nitrogen to the trees. Is this when they decompose on the forest floor or while they are growing?
Probably largely when they deposit on the forest floor. There are a few papers where they were looking at that Loberia oregon, or that lettuce lichen. I think it's probably out of the H.J. Andrews research station in the Cascades, where they analyzed nutrient input for that particular species. And found that there was some nutrient input from that lichen, because they're in old stands, there can be a lot of it. So if you have places where there's only a few cyanolichens around, it's probably a minimal input. But if you have places where there's a lot of it around, when that's, a storm comes through and it blows a lot of stuff on the ground and decomposes, all that nitrogen goes in the ground.
Okay, this group has some really great questions. Is there bioaccumulation in lichens that would threaten foragers more than ambient air contamination that might be safe at the levels which would be acceptable for human health?
That's a good question. Not that we know of. I think when, one of the big, yeah, I don't think I don't think we know enough about bioaccumulation quite yet in some of these compounds. So I think that's a good thing to look at, because that, especially when you're thinking about sort of forager resources and toxicity. That hasn't come up, but I don't know if we know enough about bioaccumulation yet to get there. Because it does, in some, it might be species dependent, because some species are better accumulators and storage species than others. And we do tend to focus our collection of, when we're collecting those bags to send to the lab, through the principal investigator, Linda Geiser, who kind of set this whole research project up, did a lot of time comparing different kinds of species of which to collect and send to the lab. And some species leach everything out really fast, and some species hold on to them for a while. So I think there's some more work to really get at that needs to be done.
So interesting. Okay, this next one. We have crustose lichens on beech trees in North Carolina that appear to be competing. Do the different lichens have methods for outcompeting other lichens for the tree surface to live on?
Yes, they do. In fact, I should have put this on the slide because I almost did. There's a great cartoon. Some of that secondary biochemistry that they have, some that we use to even differentiate them on, they form various functions, everything from sunscreens to sort of biowarfare. So especially the crustose lichens, they're a little bit more challenged. They can't kind of grow on top and interweave on top of each other like the three-dimensional leafy kinds can, or the hair ones. So they often kind of, some of their secondary chemistry is to fight for their space, so to kind of poison their neighbor. And so you'll see these things kind of trying to battle it out. You'll see these kind of complicated puzzles of those crustose lichen different species on the bark. And there was a cartoon that had kind of had a couple of species kind of battling it out. So anyway. But yeah, that's a great question. Yes, they do.
Crazy. Do microlichens at higher elevations have more sensitive effects from climate change, similar to other alpine flora?
I would suspect they do. We haven't worked with the microlichens or the crust that much, just because it's a lot more work and we don't have the capacity or funding to do it. Just because they're harder to ID and they just take more time. But yeah, I suspect because even those macrolichens, like in that graph I showed, some of those species have very narrow, sort of cold winter temperature neighborhoods that they live in, and I suspect the crusts are going to be the same thing, because they're adapted to live in those alpine conditions. And so, if it warms up, I don't think their metabolism is adapted to that.
Mm-hmm. Okay, can lichens be transplanted, and do we know how long they live?
They can be transplanted, although they actually have to be kind of in a, it has to be within their, I would say, environmental parameters. So same temp, within the temperature and sort of precipitation parameters that they would live in the wild, and kind of on a similar kind of substrate. In fact, they do a lot of what they know about lichen growth and dispersal and all kinds of things is from transplant experiments, where they actually take lichens from one part of the forest and they kind of like glue a little fish line on them, and they kind of tie them up in other parts of the forest to look at different microenvironments and things, just to kind of see, what the growth rates are when they change environments, and also, looking at how these things disperse through time in different environments. And so Yeah, they can be transplanted as long as they are in the same environmental parameters. And how long they live, I don't think we quite know that yet. On the crustose lichens, they have done a bit um, of sort of time scale with those species because they are pretty long lived as they kind of creep their way millimeter by millimeter in those very harsh environments where they have to be around a very long time to have space in the rock since their growing season's short. But there's been a little bit of carbon dating work in them, but not a lot. So there's a few species they know something about, and most species they don't really know much about how long they live.
More to learn, that means.
Yep, there's a ton of work things.
Yeah. Okay, this is from an amateur lichen lover, and they want to see them better. What sort of microscope would you recommend to them as a next step for hand lens? Next step after hand lens?
I would say I would say any kind of 10 to 40x, any kind of 10 to 40x scope that, yeah, I would just say a 10, yeah, I think they're 10, or I'm trying to remember, it's been a while since I've done ID stuff. But I'd say your next kind of, not your compound microscope, but your viewing, trying to remember, no, 1040x, I'm trying to remember what the magnification is on it, but I think just any of your basic viewing scopes. It doesn't even have to be perfect. Of course, the nice expensive ones are, they're pretty nice to see the small features, but I mean, even kind of a budget friendly scope is pretty good.
And then this person couldn't be here on time, but their question is that lichens are symbiotic, but there's a saying, they are fungi that have discovered agriculture. If fungi is getting all the benefits, could this change the concept of lichen?
Yeah, and part of the reason that I kind of had symbiosis kind of in little quotes is because, I mean, some suggest that really the fungus are just basically using and farming algae for their own benefit, right? And I think those that argue for the symbiosis is that they provide the algae with a sort of protective housing, and allows them to kind of grow in maybe more environments than they would because they're all over, through the canopy layers and things. But I don't know that we've done an analysis of, well, it's also kind of hard the free living algae are kind of hard to find on the landscape and sort of assess, but there's probably a way to do it. It would be interesting to see, well, is there, they don't seem to be harmed in the event, but yeah, that's a good question. Are they just farming them and using them for their goods? And are they actually getting anything out of that protective housing, or would they do anything on, or they would be just fine on their own? So that's a good question.
Yeah. And then this goes back to transplanting lichens. How do you transplant them?
Well, the ones that have, yeah, I've seen the experiments I've seen is just that they've tied, they've glued little fish line pieces to an end of their little loops onto the end of a phallus and tied them onto branches. But of course, these were, for experimental purposes and not necessarily trying to get, it depends on what your objective are, but it can be done. And all I've seen is, basically glued on fish line loops, tying them onto branches.
And then referring back to the microscope questions, a couple other people chimed in that I want to throw out there for those interested. There are some folks saying that 10 by 40 works great for mosses, and they would assume for lichens too. They've got one from the 80s, but it works great. And then someone else also said that a dissecting scope 10 by 40 works
dissecting
well.
scope you want, yep.
Yeah. And then let me see. Oh, somebody else mentioned into here, FYI, stereoscope or dissecting scope, often 10 by 3 or 10 by 1 magnification, perfect flower level magnification. So if anybody's interested in looking more into those are some options to possibly pursue as well.
Yeah, and and anybody getting into lichens probably wants to, I mean, I think that like the macro lichen guide has all of your introductory setup stuff, like how to do the spot test work, like, how to apply, the bleach dabs and potassium hydroxide. And there are some chemicals that are a little harder to get. But with resources, you can get them. But in any case, yeah, I would refer to that book for sure. You get that book and a dissecting scope, and then that's the first step, and then it will tell you how to do the rest.
And I think that wraps our questions for today, but we did get a few comments on really great presentation, wonderful addition to understanding the connectivity to everything. They will be watching the recording if they came late, and also watching later with their own kids and teenagers. So thank you again, everybody, for attending. Doug, really appreciate your time again. Always great to have you and sharing your knowledge on what I consider a very complex topic.
Yeah, any one of these topics could take it. Even one of those little topics could take a whole hour.
Totally. But there's so much energy and enthusiasm around lichens for sure. Okay, and then somebody says, how do you get the video to the recording of this? I will be posting it on the Cape Perpetual Collaborative YouTube channel, or capeperpetualcollaborative.org blog on Monday. Tuesday, you'll receive an email with those links that will get you straight there, or you can just do a quick internet search and search for it. So it will be available, and you'll be emailed those links. And with that, I think that is the end of our presentation today. Let me just double check that nothing else came in. Nope, I think we're looking good. Everybody, lots of thank you so much. So again, thank you, Doug. Thank you, everybody, for attending. Have a fantastic weekend and enjoy these couple of Sundays, at least if you're here on the Oregon coast, before the rain hits us again next week, which the lichens will love.
Yep, they will. Yep. Great.
All right. Thank you, Doug.
Thank you, everybody. Thank you.
Have a great weekend.
Yeah.